And our next speaker is Dr. Tom Patterson from UT Health in San Antonio, where he is the Chief of Infectious Diseases. Tom.
Thanks, GR, and thanks to the organizing committee for the invitation to speak and for the privilege of being here in person for those of us who are. It's great to see you all. So my challenge is to get through these therapeutics. The good news is there are a bunch of new drugs in development. The bad news is there are a lot of new drugs in development for these next few minutes, so I'll continue straight along, right? Here are my conflicts shown here. Most of these relate to NIH studies we've done, but also consultants at companies developing new drugs as a clinician in infectious disease and a clinical trialist. So this slide was from GR, but it shows in a paper he put together with Joanne Young, published in the New England Journal, a number of different targets for different fungi. You've heard a lot about those at this meeting, but I'll just draw your attention to in the box that several drugs really in pretty advanced clinical development with unique and novel targets do have susceptibility data that show that it could be a very attractive target for treating coccidioidomycosis in a clinical setting. And then in a number of models, including ours, you can even show that some of the drugs sterilize tissues, which has really before been pretty unknown in fungal disease. So here are some key questions for the antifungal drug armamentarium in coccidioidomycosis. Those in the grade areas GR touched on yesterday, so I won't. I will mention suba-itraconazole because when the two of us, that kind of fell through the cracks. But suba-itraconazole is super-absorbable itraconazole. It's called Tosura, is its trade name. But that's one drug that is available, and it's like itraconazole, but produces more easy-to-achieve levels. The drugs in development, though, are several. Nicomycin Z, I'm going to mention first, that John's already talked about it. Oteseconazole has been approved. There's a relative tetrazole called VT1598 that I'll also mention. Alorafilm has been touched upon by even our patient advocate on the first day. Fosmanogepix is a drug developed by Amplex and now by Pfizer, and then ibrexafungerp is also approved for treating Candida vaginal infection. There are other drugs in the pipeline, and while the cocci activity itself isn't known, they probably does have activity, and I'll show you that at the end with new forms of amphotericin, other kinds of glucan synthase inhibitors, and even the possibility of repurposed drugs. So first, Nicomycin Z. Also known VFS1 now. It's sort of a new formulation, if you will, of a drug that's been around a long time. It is first in a class of a new antifungal. It's been new for many years now, but it attacks chitin synthase inhibition, has activity against a range of fungi, including cocci particularly, but also blasto, histo, aspergillus, and others. It's selective to fungal cells, so it's really attractive. It's a subject for another day of why this has been really hard to develop. It is a fermentation product, and so that needed scale up. But the hope is that we will see clinical trials, and especially in combination, that could be useful because it has definite efficacy in murine models, and it seems to have activity in dogs as well. So the first one that I was mentioned on that early slide was these novel CYP51 inhibitors, specifically VT1161 and 1598, originally developed by Viomet, now known as a spinoff from them called Mycovia Pharmaceuticals. And it's basically a mechanism of action that's real similar to azoles. It's highly selective, though, for the fungal CYP enzymes versus the human enzymes, and so you really get a lot less drug interactions and probably contributes even to long half-life. The drug VT1161, and you see the tetrazole part is here with those four nitrogens there, right, is what makes it different from triazole, right? So it basically has minimal drug interactions and then high protein binding, so it contributes to a really long half-life. A study conducted appropriately in Violet here was called the Violet study, which was then approved for recurrent VVC in clinical practice, so it is currently available. The MICs, or susceptibility of other fungi, are shown here. Obviously, it's not the focus of our talk today, But I would draw your attention to a couple, and I'll mention this on some of the other slides. Those of you who do clinical work really realize a tremendous need for azole-resistant Candida species, especially Candida glabrata. And you'll see a teslaconazole there in the left-hand column has good activity there. The endemics are there to the bottom. You see highlighted is coccidioides. This slide was provided to me by Ed Garvey from the company. They've highlighted cocci as potentially intermediate for susceptibility, but that's a pretty low MIC. So I'm guessing that would end up being susceptible in clinical practice. You see fluconazole right next to it with an MIC on the median of 8.3. So that's one that we would generally consider what most cocci strains are like. We've had that discussion already. Clinically, I'll also draw your attention that the mold MICs for even mucorales like rhizopus are actually very low, which is also a tremendous niche need. These data were presented to stretch your memory by Lisa a few years ago at the CSG, and she gave me this slide, but basically Lisa Subitz has used a VT1161 in dog infections and showed really good reduction in those scores you've heard about earlier, and really eradication of the organism. So it really showed in a clinical sort of setting that this could be a useful tool for treating active clinical disease. If you look at 1598, it's also tetrazole. It's a different compound. It's not really related, but it is that same class. And you see in data from the fungus testing lab published by Nathan Wiederhold a few years ago, really low MICs of the VT1158 comparative to, in this series to posaconazole, but substantially better than fluconazole, which is, of course, commonly used. We also used the VT1598 in a murine CNS model. And again, now you see really close to eradication of the organism from tissues, something you really don't see with fluconazole as shown on this slide, but really substantial reduction in the tissue burden, either from tissue burden efficacy studies or even in the survival studies overall. So let me turn and spend a few moments on the erotamide, alorafilm. You have heard about it. It's the drug being developed by F2G. We do participate in the clinical trial with this drug, and so I'll share some of those data in just a few moments. But it's a potent inhibitor, as you also heard, of dihydroorotate dehydrogenase. Unfortunately, as you also heard, there are fungicidal drugs, fertilizers sort of, that will basically be used, not fertilizers, but fungicides that are being developed that also contain this same enzyme inhibition, so it's really a little bit worrisome. But one of the things, so far at least, is you don't really have much induction of resistance at all, it seems. The spectrum of activity is really pretty predictable, and many strains like Aspergillus, Lomentispora, which is highly resistant to all the antifungals we have, Scedosporium, are very predictably affected by this compound. It's also very potent against endemic organisms, cocci, blasto, and histo. And there you can see, hopefully at the bottom, MICs that are extremely low compared to voriconazole with the alorafilm in the really a very small range of drug. In our murine model of cocci, we were able to show, again, eradication of the organisms from tissues, really more effective than we had seen with any compound, and you see that pretty dramatically in this slide. Well, what about the clinical success? And John showed this yesterday, or talked about it yesterday, but just to put a face on it, and it's to say, is a complete and partial response all that we could use for a clinical response? And if you look at all those organisms, at 6 weeks with cocci, you have really almost no complete responses. But if you allow stable to be included, you get responses that are now 54%. Well, does that really matter? Here's 3 of our patients discussed, and the pathology on the right is from the second patient there, or the first patient there, who was an oil field worker. Yes, we do have cocci in Texas. We don't try to count it, but we do have it. So this guy failed multiple different therapies, And after a really short time, his PRO, his patient-reported outcome, was this the best he had felt since his initial diagnosis of community-acquired pneumonia 3 years previous. He's continued to do well. He needed to get debrided. That gross stuff at the top is his knee tissue, and you can see that yellow stuff is actually the coccyspherules coming right out of it. It was really super gross. And his knee has really continued to improve. So he's done great. A couple of others are meningitis. One woman had failed multiple therapies, including intrathecal amphotericin that was guided by Royce. He probably doesn't know that, but that was true. And her headaches promptly resolved. She's continued to do really well, but her titer remains positive. The third guy came to Texas from Washington. He's originally from Cambodia. Turned up and promptly got coxy, wasn't diagnosed. He had a super high CSF titer for IDCF. He started on standard therapies, didn't respond, ended up with a bad spinal meningitis and a C5 quadriplegia, so really horrible infection. He turned up to us and we started him on olorofilm. He promptly had a better mental status, he improved arm movements, and his CSF titer decreased. Obviously, he didn't improve a lot, but I would say all three of those are pretty successful therapies. If you look at the study that Johan Martin presented at ID Week, you see that same sort of thing. Compared to historical controls, the average duration of no symptoms after many years was almost none, and in the olorofilm group, you really got a good response. So, I think there are data. So quickly on the last few, one is the Amplex drug. It's a drug that's now being developed by Pfizer. It inhibits the enzyme as shown there, the GWT1, and is a prodrug, so it's also available IV. You can see that the susceptibilities for coxy are also extremely low with this compound and also active against fluconazole-resistant yeast. In animal models, we've looked at it a number of different ones. We haven't looked at it in coxy, but we have looked at it in other invasive infections. There is a phase 2 trial in progress, and the drug is known as phosphomanogepix. There is one study that Josh Fear and his group published that showed good activity. They used high doses of drug along with the cytochrome inhibitor to produce those decent levels in mice. And you can see really that outcomes were good with a significant reduction in the overall organism load. A couple last slides. The amphotericin cochleates is kind of a cool idea. It's amphotericin bound up in little cochleate balls or nanoparticles. It's called MAT2203. Really interesting data from Africa that's coming out now, the ENACT trial, that looks at the drug compared to conventional amphotericin, produced good survival in cryptococcal disease, really hard to treat and really important in Africa, with really good early fungicidal activity. Could be a pretty cool oral idea for coxy in the long run. Ibrexafungerp is a Synexis drug, also approved for vaginal candidiasis. It's a glucan synthase inhibitor, and that drug also is one that's being looked at in phase 2 trials for Candida auris and other resistant fungi as well. Rezafungin, for the last couple of ones to show, is a drug that Dr. Thompson, GR, has used, been a primary investigator for Candida, but it's also a long-acting injectable drug, so it could be one that even for IV therapy could use in an infrequent basis, but there are really no data at the current time for coxy per se. Then what about repurposed drugs? Repurposed drugs are kind of cool. One is people thought about is sertraline, the antidepressant. It may prohibit some kind of transport differences. It does have in vitro activity in coxy. Unfortunately, it also does in crypto and didn't really work in the clinical setting, but the MICs are not unreasonable, so these repurposed drugs are probably worth looking at as well. So I think to summarize, new antifungals are clearly needed for unmet medical needs, and hopefully I've shown you in a quick whirlwind the ways that new compounds are being developed. This slide shows our supporters. Thanks to Donald Love and NIH for support that our lab has had for coxy in animal models and in new drugs, and also in the Unite teens that you've heard about as well. With that, I will stop and thank you for your attention.